I'd like to begin the morning by introducing Ranga. He's the PhD scientist and the Chief Scientific Officer and Senior Vice President of Clinical and Translational Sciences at BERG. He oversees all research and development, strategic planning, and implementation of workflows for effective translation of discoveries through pharmaceutical and clinical development. He is also responsible for a robust pipeline of assets in multiple disease indications, from validation of novel disease-specific targets to drug discovery and development programs moving into IND-enabling proof-of-concept clinical programs. Dr. Sarangarajan is an inventor and architect of the BERG interrogative biology technology, currently focused on next-generation R&D innovation in the integration of artificial intelligence-based analytics, bioinformatics, and for speeding up identification and validation of targets, tractability, drug discovery, and pharmaceutical clinical development. So welcome, Ranga. Without further ado, I'll let you take on the stage and begin your talk.
Hey, thank you, Devanu, for the kind introduction. I'm going to start sharing my screen. Please let me know if the audience is able to see.
Yes.
Excellent. Good morning and good afternoon, depending upon the time zones. Just a brief summary. So basically, BERG, we are a Framingham-based company, clinical stage with multiple assets, but our main area of expertise is the leveraging of human-based biology as a source of our data in identifying targets and then developing those targets for in terms of validation and drug discovery. And we, over the past probably 11 years, have invested heavily in the use of AI and machine learning, starting in about as early as about 2008-9, when AI was not the mainstream or a buzzword. So today, what I'm going to talk about, I was struggling in terms of the topic itself, and then finally decided to use a bona fide case study from our internal data structures and stuff that we have actually done, and share with you in terms of use of biological information and its overlay in the context of high density data sets, and trying to leverage the information that comes out in the entire lifecycle management of an asset. And I thought that fitting a needle in a haystack is very appropriate in this setting. And so, without hopefully, let me see, it's not moving. Okay, so just to focus on a case study, I decided that it was best to concentrate on something that most people would have heard of in terms of the API. But at BERG, we have something called a program that is focused, a formulation called 31510. So BPM 31510, we have a fairly robust clinical programs. These are the program itself. The API has been formulated as an IV infusion, as a topical formulation, as well as an oral. And each of these have different clinical development path. But most importantly, what you would be surprised to see is the fact that 31510 is a formulation for Coenzyme Q10. And when people first hear this, they say, okay, you know what, can you go to the pharmacy, get an OTC, and actually use a benefit? Why would you want to spend time and effort developing it with the kind of price tags within the pharmaceutical industry? What is fascinating is the fact that some of the early work that was done with this molecule in our labs, we were able to demonstrate that coenzyme Q10 at very low concentrations is able to effectuate an anti-cancer effect. And when we looked into the literature, we had a tough time finding a formulation that would suit the purposes to reach the kind of concentrations that would be required. So did a lot of work, and eventually were able to generate a very stable formulation of CoQ10, which is an oxidized form. And I'll get to that. Please remember, the oxidized form of coenzyme Q10 is important. And when combined in a ratio with lipids and surfactants and other ingredients, we get an extremely stable formulation that at 25 to 30 degrees, you can keep it at room temperature, and it stays as a nanodispersion of 40 nanometers on an average particle size. Now, the reason for specifying the oxidized coenzyme Q10 is the entire world, if you look at the literature, it really talks about the reduced form of the coenzyme Q10 as being the antioxidant, which in fact we have significant data. We probably are one company where we have the largest granular molecular profiles of human-based biospecimens treated with coenzyme Q10 at various concentrations and comparative controls. So the formulation itself, just to give you a quick background here, the formulation, given that coenzyme Q10 has to get into the mitochondria, in the top panel, what you're looking at is a mass spec-based validation of accumulation of coenzyme Q10. So the formulation is able to deliver very high concentrations. And the second aspect of this formulation was very interesting, where when we did the biophysical assessments of it, the primary cells do take up a little bit, but the accumulation within the cancer cells is significantly higher in order of magnitude. So we know that we were able to deliver it into the cells and very specifically into the mitochondria when you compare the whole cell to the mitochondria. And the final piece was this interesting and exciting fact, and this is a very small subset of the data that we have internally, where when we treat cancer cells, we see a dose-dependent decrease in cell viability, and we don't see any major effect on cell viability in the normal human cell lines. And so that was one of the earliest set of data. So then the question becomes, okay, so the general understanding of coenzyme Q10 is it's a molecule, goes into the mitochondria, it sits on multiple membranes, including the plasma membrane, but its primary role is as an electron transfer component within the electron transport chain of the mitochondria for priming to generate ATP. So how does CoQ10 kill cancer cells? And why is it not having the same effect on the normal cell? So literally, when you coming back to the understanding of coenzyme Q10, just to give you a brief summary here, this is from the Linus Pauling Institute, where they have a significant database on coenzyme Q10 research. Coenzyme Q10 sits at the intersection of the complex 1 and complex 3 around the complex 2 binding site, and basically is responsible for moving the electrons across the electron transport chain in the inner mitochondrial membrane. And then there are the substrates that are turned over to generate ATP synthesis. So that is pretty much the fundamentals of CoQ10 role in biology that you can get. And if you go and look at the literature, and trust me, we have mined through this ad nauseum, there are various facets of its benefits in the human biology. Everything from treating cancer to normal biology, cardiovascular health, immune system, you name it, and you can find references. But to date, if you start getting into the actual MOA or the mechanism of action of this compound and how it performs within each individual cell type, there is a huge variation in terms of the scientific literature in highlighting what a clear path is for it to effectuate some of its benefits. And so we were looking at it from a cancer perspective. So we were able to generate a formulation where we are able to achieve supraphysiological concentrations of CoQ10 to not only the target tissues, specifically in the cancer, but also in the mitochondria. We did it in the cell, in vitro cell model like everybody does. We have done this, we were able to reproduce it in xenograft models, organoids, and so on. But the bigger question was, knowing what was in the public literature, how do you fit its role within the mitochondria into an anti-cancer paradigm? And that's why, so the coenzyme Q10 MOA as we know it was the needle, and how do you fit that? into the complexity and the plasticity that one encounters within normal biology, and how does that differ within the cancer biology. And so it was something that started off 10 years ago. And happy to say that although, the 31510 was one of our first test cases for the use of BERG's interrogative biology platform to understand this in a data-driven manner, since then we have approached this, we have used this method to tease out the biological underpinnings of a significant set of other compounds. So coming to interrogative biology and the BERG platform, I'm not going to get into too much details here, but just to suffice, what we start off with is human-based biospecimens, because coenzyme Q10 is endogenous to humans and some of the higher order animals, whereas when you start getting into the rodents, it starts getting into Q9, Q8, and so on. So interrogative biology is a process where we are enabling the comprehensive phenotypic inclusive molecular capture of human biology. And what does that mean? So, the traditional conventional drug discovery and development, right? You have a hypothesis, you generate the models, you try and validate the null hypothesis, move on, and so there is a linear fashion. Where we were able to turn this on the head was by using a data-driven approach, where we said we didn't know anything about coenzyme Q10. What is it doing comprehensively to all the various cell types that it encounters, whether it's a normal cell or a corresponding malignancy, and start generating large amount of data sets, everything from genomic, proteomic, lipidomic, metabolomic, as well as functional information related to a specific molecule readout. In this case, it was more from a mitochondrial function as a phenotype. And then we started implementing the use of various, we started test driving some of the mathematical formulas in terms of that was available in the 2008-9 era, and eventually developed a suite of artificial AI-based algorithms to parse through the data. And so what we were able to then achieve was creating a full biological narrative, not only for coenzyme Q10, but now we do this for almost everything within at BERG, starting from the early discovery all the way up to clinical development for almost all our assets. And it starts with the patient samples. You generate the high density data, biological data, meaning the lipids, the metabolites, the proteins, genes. But what we combine it with is, if you look at it from a holistic manner, there are two separate independent parts of data collection. One is the data collection, which would be from an individual at the clinical side of it. So in the everything from demographics, electronic medical records, as well as the extracellular fluid-based data capture. So you can get molecular high profile or high density molecular profiling from plasma, Buffy coat, so on and so forth. But that's not where the causal effect is present. So in parallel, what we are also doing is getting biopsies, healthy normal cells, as well as organ cells from organs from diseased patients, put it into the wet lab, as well as do perturbations to capture the molecular plasticity between normal as well as the disease environment. And then we connect the dots between the real world data that we get from the patient, as well as their corresponding molecular profiles from the organ systems, to generate a holistic map, which is what the full biological narrative is. And we did this with coenzyme Q10. So what we have here is just a framework, right? On the top, we do have the in vitro cell model where we did the perturbations, and especially the cancer cell, the hallmarks of cancer is very well described in the public literature. So everything from modulating glucose to all the other parameters, you can capture a very real comprehensive molecular profiles. And correspondingly, we started sampling things from the human system, literally developing biobanks through prospective clinical trials and getting access to patient biopsies, FFPE samples, so on and so forth. And after the molecular profiling, we run it through something called BASIS, where that's the proprietary software at BERG. And it enables us then to identify novel targets as well as look through the disease target enablement for clinical development. So in this context, what you're looking here is an actual data readout from BERG. So on the left-hand panel, what you're looking at is extracellular acidification rates and oxygen consumption rates, which are two phenotypic functional endpoints that are part of the model development in the in vitro study, as well as when you move it towards certain in vivo components, especially in xenograft models, you can actually take the xenografts and capture OCAR-ECAR values for those tumor from the organoids as well as the xenografts. So you can generate that kind of high density functional data and embedded within the molecular profiles. And that's what you're looking at on the left-hand side. On the right-hand side is the entire clinical data set from almost 2,000 patients who have been exposed to coenzyme Q10 at some point during our clinical development. And this contains everything that we have. So this is, it looks like a hairball, but as we start digging deep into this, you can actually start teasing out the specific connections using our Bayesian AI algorithms. So then the question was, how do we fit this proverbial needle into the high density network, which is basically a haystack? So we actually went around doing this systematically with not only in our preclinical discovery model, but then independently start overlaying the molecular profiles within 104 patient phase 1 all-comer solid tumors. And then we have now moved to phase 2 randomized GBM trial as well as pancreatic cancer trial. And so the pancreatic phase 2 is done, the GBM is ongoing. So we are using these molecular profiles to actually run it through clinical development. But the big question is, coming back to what we started off with the presentation, what is it telling me about coenzyme Q10 and the anti-cancer effect? So what we did was we used a combination of AI and ML and a slew of other computational capabilities that we brought in to specifically look at the molecular profiles in a very systematic manner, but in a data-driven manner. And then started looking at what are the highlights for the mechanism of action, or what is representative of adverse events, and can we actually identify markers from this data set on patient stratification between responders and non-responders from the phase 1 to the phase 2. And I'm happy to say that this works like a charm, at least in our hands. But coming back to the story towards coenzyme Q10, just to give you, for those of you who may not be aware of this, cancer cell metabolism is a big thing. It was considered to be it was added as an additional hallmark of cancers a few years ago by Weinberg. But basically, it is a switch of metabolism from a mitochondrial-centric generation of ATP towards a non-mitochondrial glycolytic-driven ATP, which and I have to say this, that it is not universally adapted by all cancer cells, because some cancer cells have a combination of both normal mitochondria as well as the Warburg phenotype. So it's a whole range of it, but this is just to give you a brief summary of the fact that for sustained growth in cancers, cancer cells tends to short circuit the mitochondria and generate predominant ATP from a glycolytic pathway. And so when we started generating the high density data on the cancer cells and started comparing it to the normal cells, a few major pathways got hit through the analysis. And we were able to identify that CoQ10 or 31510, as far as CoQ10 is concerned, hits metabolism in 3 major paths. One is it is doing it at the level when glucose enters into the cell, into the cancer cell, especially in the PPP shunt, as well as certain specific pathways within glycolysis. It also impacts certain very specific steps within the Krebs cycle, within the mitochondria. And at the level of the complex 2, complex 3, it because of certain physiological push, because of the excess CoQ10, there is a significant impact on the generation of ROS. So basically what CoQ10 is doing is when you deliver supraphysiological concentrations to the CoQ10, it is seeming to push the cancer cell across the apoptotic cliff. And that has Tony Letai has been working on that concept and the BCL2 superfamily of proteins for a very long time. The data that we generated actually was very complementary to that. And what we were able to figure out is that this is very consistent. And here I'm just giving you a very brief summary. We have done it for innumerable amount of cancer cell models, and consistently what we are seeing is the reason why I put this particular format is the black profile is a representation of cancer cells that are extremely aggressive. So those that are aggressive, fast-growing tumors, you see a higher percentage of ROS being generated with coenzyme Q10. And that, again, the oxidized CoQ10 or 31510. In slower-growing tumors, you still see an increase in ROS, but it is it's much less prominent in the initial stages, but eventually it still manages to activate apoptosis. But in the normal cell, interestingly, at even very high concentrations, you are still seeing a decrease in ROS. So there is a differential that the human cell is able to self-regulate itself in terms of the excess CoQ10 that it sees. And here is the just a proof of concept again. Once just to show that 31510 here, I'm showing it against a pancreatic cancer. We have published recently on both the pancreatic as well as GBM and some of the other cancers, and more publications are coming out. They are in various draft forms, but consistently, supraphysiological concentrations of coenzyme Q10 in the xenograft models and in the clinics is associated with reduction in the tumor size, as well as especially in the xenograft model, it kills the cancer cells. So then coming back to what is the mechanism, and this was very interesting and surprising for us. So like most people, right? Once you get the readout, you want to see some of the things that are very well established in the literature, but how does this perform within what other people have published? And this is an example of a cancer network from a public domain. For obvious reasons, I can't name where we got the source, but within that, when we overlaid our data, and that's why you see all the other dots have been hidden here because I couldn't disclose this. But the three major hubs that we see is the differential regulation of p53 pathway and the increase in BAX and decrease in BCL2, which is classic. And this is something that we have seen consistently across all our cancer cells. So in short, we started off with a molecule, threw it into cells, generated a large amount of molecular data, used AI and other ML to then generate these networks, and then started profiling and backtracking as to the mechanism in terms of overlaying the data set into the actual clinical piece and started figuring out what the mechanism is going to be. Then the question becomes, okay, so now that we started teasing out the MOA of this, what we were finding out is that although ROS and activation of apoptosis is consistent across cancer types, how does the cancer cell reach there is completely different and varies accordingly. But more importantly, what we wanted to figure out was if there are additional insights that can be gleaned. And here is a classic example where we identified PDIA3, and again, this is public knowledge now, as a modulator for ECAR, OCAR, which is in the model. But what you are looking at is PDIA3 is from our phase one solid tumor trial in terms of response. So increase was associated with a good response compared to the baseline. And so we were able to identify a biomarker that is actually potentially predictive of response to 35-510 in solid tumors. And now there are additional markers in the panel that we have identified and are in the process of validating in our GBM and pancreatic cancer trials. So I'm actually showing you some interim data here, which we recently presented last year at ESMO, where in the phase 2, we did confirm 2 markers that are differentially expressed between normal and cancer. Again, this is just a serial effort where you collect the data, you collect the samples, generate the data, run it through the AI, and then independently validated. So the phase 1 trial was overlaid into the phase 2 trial, and we identified these markers. So I know I'm coming to the top of my time left. So hopefully, I'm giving you a good window in it may not be convincing, but at least, hopefully you get the rationale on how we move this, because this is almost 10 years worth of work. But what we have gotten out of this is that coenzyme Q10 acts at the level of the mitochondria. Okay, that was not a surprise. But oxidized coenzyme Q10, the pro-oxidant mechanism, is what is really responsible for the anti-cancer effect. And this has, we have shown this, and that was also basis for patents and everything else. But what we were also able to achieve in this process was that certain lipids are natural partners for coenzyme Q10, and that helps in generation of formulations that is extremely stable and deliver high levels of CoQ10 into the biological systems, especially in the humans. Believe it or not, we have an IV formulation that is currently in development for the solid tumors, pancreatic cancer, and GBM, where the concentrations achieved is in excess of 200 micrograms per mil in the human patients at Cmaxes. So we know that it is extremely well tolerated with almost no adverse events reported in these patients. But what was more important in this exercise was the amount of data that we have accumulated over the years has provided us with very amazing insights into what CoQ10 does, not only in the cancer cells, but in other human tissues, from the heart, the kidney, immune system. And so we are parsing through this data. Like I said, some of these are already in print. I think 2 or 3 of these manuscripts are already published. We are in the process of mining through this information towards to add to the value. So, just I'm getting into the summary slides just in a minute or so. I'll end here. But basically, the paradigm is generating, this wouldn't have been possible even 15 years ago, but we are at a point where technologies, high throughput technologies, provides the opportunity for generating massive volumes of data. It is important to figure out what kind of data you want to collect, and then put the technologies and put the resources to actually do the data capture. And hopefully, I've given you one method of using the data accumulation paradigm in both a discovery as well as clinical setting, and connecting the dots for us to make data-informed decisions, because that's where the future is headed. And for the CoQ10 storyline, here is where we are. We are closing in on phase 3 designs for some of our trials for coenzyme Q10 in solid tumors for specific indications, and we have used molecular profiling and the use of AI machine learning tools in almost all the major bullets that you see here to help us navigate through and make data-driven decisions for our clinical assets. And this is just one of the many assets that we have done this. And so I'm going to leave you with final thoughts and open to questions. All of us on this call realize that the drug discovery and development, the pitfalls are immense, and it can be extremely time consuming and expensive. if there are failures. So anything that can help us in navigating through this minefield is going to be extremely useful. And this is my personal belief that interrogative biology, the way we approached it, is a major process-driven asset that we have developed over the 10 years that is extremely useful in walking through this minefield, so that we are running our discovery right through the development in a data-driven manner, and also providing a comprehensive pathway for de-risking assets so that we can get more hits on the goal and success in the clinical development. I want to thank all the organizers for giving me this opportunity to speak, and also the participants and the audience for listening. And I'm open to questions.
Wonderful. Thank you so much, Dr. Ranga, for your brilliant presentation. It's quite mind-blowing, as you mentioned, the progress from drug discovery to clinical trials. And if there's any questions for Dr. Ranga in the room, you can type it in the discuss or in the Q&A tab to your right. Otherwise, Dr. Dabanu, do you have any questions for Ranga?
Saranga, a great talk.
Thank you.
One question I have is, so I think it's amazing what you pointed out, that what you were doing in terms of ML and analytics was far before the current buzzword stage of where ML and AI is, right? And a lot of us who have been doing computing for a long time realize this. So where do you see this heading in the next few years? Is there a lot of hype? Is the hype going to bubble out and get back to basics and reality of what things should be? And where do you see ML AI going in future in the whole discovery, repurposing, biology discovery phase?
Yeah. So again, I may get some flack for this from the audience, but I think we are in a data overload phase right now. There is too much data floating around, and so when people don't realize is that data storage is expensive, and then the cost of goods in terms of the return on investments from a health perspective, given all the expenses that one encounters just on the baseline asset development in biology. So when you add that to the data piece, then it starts getting exorbitantly expensive. And so, I'm a firm believer that every outfit needs to figure out what kind of data that you really need and go after that, and then figure out the fundamentals of AI and ML that works for them, because every math is different. And so I think it's no more a hype in my view. There is hype in certain components of it, but people are going to be successful if they are able to reproduce their use of AI and ML to consistently show success. And then the other ones, obviously, the failures are going to drop off. Just putting it into perspective, right? When you hear about things like Watson and all, maybe Watson was too ahead of its time when they started doing this, because if Watson or things like that start coming up 10 years from now, where NLP is going to be just a small fraction of the amount of data that is going to be integrated for meaningful informed decision support, then we are going to be looking at a completely different paradigm. And so I see that data is going to get streamlined. Data, the fair use of the fair standards and some of the interoperability questions that folks are working on. I think this is all going to get consolidated into few successes, and the rest of the field will just have to be, there is still going to be a need, and there are going to be use for these niche AI ML pieces that will then feed into some of these main data trunks for everything from discovery to drug development, including post approved lifecycle management of assets. Because I think the distance between development of a drug towards management of health, that divide is closing very rapidly. between the provider and the actual developer of a drug. And so I see, this is going to explode, and consolidation is going to happen over a period of time.
Wonderful, awesome. Again,
Thanks.
Dr. Ranga. We thank you for your brilliant talk. And again, if anyone has any questions after the event, you can email myself and our team. We can forward it to Dr. Ranga and his assistant, Leanne. And with that said, we will be now heading to the next session with Nitin, a systems biology approach to drug discovery. So Dr. Dabanu, if we can head over there.